I thought this was a sponge from way back.
All right. There may be a sponge on that rock over to the upper right. There's
sediment.
It's probably my eyes, but it looks like something's there. Or is that just sediment?
Sediment. There's a black thing on
sediment,
that spiral coral.
Come on in.
Jellyfish, I believe.
Where are you seeing it?
Bottom
side. Oh, it's a benthic.
Oh, yeah, we saw this earlier. This is one of these carnivorous jellyfish.
This gives us a different side of it.
And the genus
It's actually
Agina.
hanging on. I'll let you get settled.
So do these guys just float along until they encounter a coral and then munch away?
Well, that's our best guess.
Get a little bit left.
I don't know, as I mentioned earlier, when we saw the first one, I'd never seen this type of animal before. That was in 2015. I think Scott may have been the one that sort of tracked down what this thing was.
Yeah, I think that's right, Chris. I should probably look it up again to be certain. I got some feedback from Steve Haddock at the Monterey Bay Aquarium Research Institute, who does a lot of work with gelatinous zooplankton. And again, this one...
Yeah, it looks sideways, Scott.
He's hanging on from some tentacles, but it looks like the mouth, the manubrium, is that darker structure hanging down, so it doesn't appear to me that the mouth is over the polyp. But it does seem to be hanging on to the branch with two tentacles. This is beautiful, beautiful imagery. Really nice that we're able to sort of move around this, or else the branch is cooperating.
So, Scott, it looks like
we get various angles. That's great.
It looks like there's an adjacent branch that has missing tissue. You can see it right there.
Yeah, that's a lot for that to eat. I'm not sure he could have eaten that much.
We're talking about a jellyfish hanging onto a coral to begin with, so just saying, just might be taking little pot shots at it.
Well, it could have been like a...
Maybe once it, it squeezes the bell, the umbrella of the jellyfish to swim, so maybe it hangs on to one branch and swims over and can nibble on the other one. Let's do all kinds of speculation while we're looking at this.
So what do you think local up is?
Wow, that's a lot of rubbly substrate up there.
I would say anywhere from due north at 25 degrees, yeah.
Looking at D2 sonar, it's looking like north.
We're going to get one a little higher up.
Copy that. Yeah, I'm at 10 right now.
We've
So
gotten one already in the
we're
bottom.
taking north. Is
What we're lacking is biologics. A little snail.
Snail.
Yeah, we can just start going north. Nav. I don't think that'll be an issue.
Sort of looked like he's lost a portion of his shell.
No, it's just sticking out.
Okay, that's good. I think we can move on.
And we're continuing to move up slope along the flank of this volcanic cone. It's on the side of this guyot. I think it's volcanic, that is.
Co-pilot, let me know when you are ready for another.
Yeah, pilot switched out about 4.5, 5.0, so I think we're in good shape for another move.
Great. Bridge, nav.
So we think this cone is a product of what's referred to as secondary volcanism, which means...
We'd like to repeat that same move, 30 meters, bearing 010, speed 0.2.
The initial event to create the entire seamount.
Range 30 meters, bearing 010,
That's
speed
apparently
0.2.
how you can
Good copy.
You sometimes get these small cones forming on the top of old carbonate drowned reef terraces. And if that's the case for this one, it happened a really long time ago, probably not that far in time from the original seamount construction. It obviously had to sink down below the surface, below where a reef could form before the second eruption. But this is a lot of manganese crust, so many millions of years ago, this cone was formed.
ROV nav bridge, move initiated.
Thank you, Bridge.
How's the tether been looking, Sean?
Good. I haven't seen any signs of any kinks forming. I
There's
think all's
something coming
well.
off the top of that little whip.
We
Yeah,
turned
coming in,
to starboard this morning, so we're in the same configuration we were on deck.
Copy.
And it's a brittle star and a dead sponge stalk, it looks like.
Something
Yes.
down here. Coming
There's
out,
a,
thank you.
there we are, a crinoid. Or feather star.
If we could pan up, I think I saw another one of those. Yeah, in that little sort of pit there, right at the base. Looks like there's a little sediment structure. I think that's another one of those protist tests.
You're infatuated with those, aren't you?
Oh, sure. Why not? Who wouldn't be? They're giant killer deep sea amoebas.
Well, if you put it that
Well,
way,
I mean,
it's interesting.
detritivores, maybe deep sea
Right.
detritivores, but I know that's not as exciting. But
Yeah, you almost had me interested in them in there for a while. Well, I'm interested in them anyway, but
well, I was just reading about these. Thanks to a question asked by Dave the other day, I was about whether or not a larger type of amoeba might actually have more than one, or a larger nucleus, or if it had more protoplasm.
Squat
or
lobster.
And
Get rid of
it turns out
lasers.
that
Small
a lot
pink.
of these larger sort of amoeba-like
Lasers.
amoeba-like organisms have more than one nucleus. So
Really?
yeah, so
Wow.
it's unclear
You
how
want it?
much protoplasm is in there. It's quite a lot apparently, but to sort of help with the regulation, they're multinucleate. So funny things.
okay, they're more interesting than I thought. That's pretty interesting.
Well,
Multinucleate
Oh yeah, well, there's
huge.
I mean, they eat animals. So here's another crino.
proisocrinus.
Yeah.
Might start.
Discovered.
Yeah, you might feel a tug too.
Chris, I would say that in that respect of having a multinucleate cell, they're similar to the glass sponges, which are also syncytial. Essentially one giant cell draped over a glass skeleton
Go ahead
that
and
has
roll.
multiple nuclei.
See,
Thank you. Yeah.
here's an acorn barnacle, deep sea acorn barnacle of some kind. These are more reminiscent of the barnacles that one sees at dock harbors and that sort of thing.
Yeah, coming up.
So pilot, how's the current flow right now? Is it pretty quiet, or is there any current detectable?
Yeah, there's definitely current. It's going to the northeast, maybe.
East northeast.
So it's blowing me. Yeah, it looks like it's blowing me. Yeah, east northeast in that general direction.
So again, that brings up or begs the question: what is wrong with this place? You have hard substrate all over the place. You've got good current flow. You've got elevated terrain off the top of this guyot, so very little sedimentation. I don't know. In comparison to that incredible,
There's on the lower
sponge
right.
thing.
There was a rock pen.
Hey, coming in, Roland.
So, Pilot,
Go ahead.
let me just throw this out, but I wonder if there's some interest by the group in collecting one of these. It looked like it was in a pretty good position. I can throw this out, but somebody the other day thought this would make a good collection.
I think we'll be okay here. I don't think we need to move back. If you
does anyone think that this rock pen would be of
copy that.
interest?
All bio boxes are available.
They aren't often collected. Sometimes they're a little difficult. This one's got a pen running the length of
ROV Bridge, move
the
complete.
stock there.
Thank you, Bridge.
Therefore, it might be pretty easy to grab. Some of them.
Go ahead, Dry Lab.
Yeah, some of them.
Yeah, local time off bottom
aren't that
is
easy to grab.
15:45.
Sort of retract up, but we haven't seen tons of collectibles so far. On the other hand, Scott, have you seen any bamboo corals that you think would be particularly interested? I'm open for that.
Look at the little,
we've already collected Victor
things
Agorgia.
inside the tissue on the bottom there.
There's possibility of getting a sponge that had those weird shrimp in there, but just throwing it out.
Well, one thing I can back you up on, Chris, is that we know this is a rock pen, but the only way to actually know what species of rock pen it is would be to collect it. And you'll have to help me. My memory isn't good enough. I don't recall if we have attempted to collect a rock pen during Capstone. We have certainly seen several, but I don't know that we've collected any.
I think Tina is supporting this.
Pilot,
She said she's not
rotate
sure if
keep you in view.
Phil included wine
copy.
material, and she thought it was more
Co-pilot,
interesting.
do you want us to move back?
This
No, I think we'll be
straight
fine.
form, which
I
I
just
think
want to
this is what she's
rotate.
saying. This is more interesting.
To keep in view.
That's not correct.
I don't see anything dangerous ahead, though.
Asako can't see her video right now. She's not connected to it, so she can't really weigh in on this. Pilot, why don't we go ahead and do this? We haven't collected one of these, I think.
Copy that.
I'm not sure if we've
Chris,
ever...
So if you are going to collect, it'll come off the bottom extremely easily. And what I would suggest is a little bit of a delicate touch, so it's not cut, but just sort of gently held onto and lifted off the bottom. It's going to be very easy to come off the bottom.
I assume you copied that, Pilot, right?
Yeah, I copied that.
Okay.
All right, let's get both boxes ready to go, Sean.
Yeah, copy that. Both are ready to go.
No. Yes, it is. We got the rock.
Do you want you up here?
No, I'm good.
Copy.
I'm probably going to go port here.
You got it.
Doesn't want to come off.
Pilot, would I see maybe bumping the base a little bit? Just the slide. It's like a suction cup.
Yeah, copy.
Are you doing okay, co-pilot?
Yeah, co-pilot's happy.
Great.
Want some zoom on HD1?
Doesn't want to come up. Well, I might just have to grab it and tug.
Yeah, and I think that'd be fine if you can grab it as low as possible. And if it separates, it separates.
You've got it. It's off.
There, I think it came up. Oh, very good.
Drawer is clear.
I thought it was going to make a lyre up,
drawer.
but I thought it would just fuck off. But it was starting to look like it was being recalcitrant. So I just want to say it's nice that we were able to pluck it off with the peduncle, because in some of these species, the sclerites are only known from that peduncle. There are no sclerites actually in the polyps itself.
Oh, I didn't know that, Scott, so thank you.
Yeah, that's just in some of the sea pens.
All right, tracking. Nice job, pilot.
Hey, thank you very much, pilot. Excellent
You're welcome.
collection.
Okay.
Oh, happy? All right, sealing valve packs. Pew off. No, pilot, I did have to rotate a good bit to keep you in view to starboard.
All right. I'll come your way.
Copy.
Nice job, Sean. Thank you.
Oh, you did the hard part. Yeah, if you just want to keep coming what you're
Okay,
doing.
continuing up. We are currently at 1,500 and about 12 meters. Come up from about 1,600 meters, so we're still climbing the flank of this volcanic cone. Oh, look at this. Oh, it's just a proisocrinus. Got all excited. And is that just another Iridogorgia? Just another Iridogorgia?
You took the words right out of my mouth.
All right, Pat, that looks good.
There's just another little bolosoma sponge. I was trying to be fair.
Don't leave out the geologist.
Yeah. Oh, that's right. And there's just a few more rocks here.
Here's another one of those little Egina jellyfish, though.
That's the exact same one we imaged before.
Oh, is it?
I don't think so.
Yeah, we could perhaps pass on this one. Yeah, it's just caught up next to it. Or another one caught up
This
next
is
to
a
it.
new one.
Yeah, and it's holding on.
But it's that branch is somewhat clear.
Aha! See? That's nice.
Yeah.
Man, it's like we came back to the same colony almost, isn't it?
Well, that's what we thought at first, and then Josh, the pilot, recognized this is different. The polyp's still there at the
This
end.
is fantastic. I mean, this is the value of the Okeanos Explorer program right here, that we're getting to see more of these observations. The more observations we make of a similar nature, the better we can refine our questions and our hypotheses and try to understand what's going on in the biology.
It's probably the best image we've ever gotten of one of these, maybe because the Iridogorgia is elevated, so you can get closer to it with the camera. But that's extraordinary.
You know, it's interesting. The one next to it, the branch next to it, is also denuded. It makes me wonder if it's using those arms as kind of a sort of a way to transfer itself from one feeding spot to another. That would be interesting.
Kind of swinging like a monkey through the trees, right?
You read my mind.
Yeah, so it's really neat. When in 2015, I think each time we saw them, the subumbellar surface, or where you see that tube sticking out, pointing towards the bottom, the mouth would be on the end of that little tube. Those were over the polyps. So it's only these last couple of observations that I have seen them hanging on with the tentacles. So great stuff.
I think
something this suggests to me is that when this jellyfish encounters a coral where perhaps it's feeding, it doesn't want to let go and go swimming off back through the water column. Why not just hang on until it's digested its meal and then start feeding again? Same sort of thing I imagine the sea stars are doing.
Yes, that's a good notion. The inside is also quite plump right now. We could probably tune into another one later and see if the little gastrovascular cavity actually is emptied or kind of full.
Do I see this small sponge, Chris?
Yeah, let's do a sponge.
Yeah, come on in.
This is also fantastic because Les and I have often wondered, what is it that's getting to the tips of these branches that are way up off the bottom? And it looks like it must be something fairly delicate. And so there you go. That could be the culprit.
Yes, absolutely.
All right, Roland.
So it's not always the echinoderm's fault.
Yeah, so this is a bolosoma. Got a couple of these. They're very small. We don't have any of the big bolosomas that we've seen on some of the other dives this cruise. So we're slowly getting up to an area that's steeper as we get closer to the cone, and I'm sort of interested to see how, as we get into steeper terrain, whether we get a little bit increase in the number of animals. We're looking at a dead coralliid stump here, so that was a chemicorallium at some point.
There was something on the rock face just behind it. Could we zoom in on that?
Come back in, Roland. Where are we looking?
So above the lasers, 3 rocks up, and there was like a white, like
I think
just
it's one of those anemones
right.
again.
Yeah, so just pan down just a bit.
Taking that thing?
The one to the left, actually. There's like a little, is it a worm? Some kind of scale.
Oh, yeah.
Maybe a scale one.
Okay, it might have been one of those same worms we saw earlier today. Huh. Interesting. Yeah, okay, we can move on. At least it was a thing this time. Wow. It's a big broken vesicle of magma.
I think this is another Metallogorgia pilot, but let's go ahead and take a look at it. Oh, okay. There's a bamboo first.
Oh, is this what you're... Oh,
no, I was talking about the one to the upper left. I didn't see this. My guess is it's the same one. Scott, is that your guess too?
Ready for another co-pilot?
Yeah, sorry, I was just typing a message there to Steve Haddock about these jellyfish observations.
No, I think we get one in.
Miracle will tell us
probably at 5-0.
Really quickly throughout the scientific world of these cool observations.
Nothing in front of us that's concerning, so I think we can get another one going.
Earlier in the dive, note that the
another 30
needles are fairly prominent and protruding through the tentacles, so this is different from the B1 or B-clade
bridge nav
coral that we were looking at earlier. This is something different.
It's the bridge, go ahead.
So
Another move, please. 30 meters, bearing 010, speed 0.2.
Maybe something else. We'll have to go back and do some comparison.
Range 30 meters, bearing
So how would you like us to
010,
annotate this, Scott, just to give you a clue what it could have been? Would you
Good
want
copy.
an I4 clade, if I'm not mistaken?
Yeah, let's put question mark clade I4.
Okay, thank you.
Right,
That's exactly how I will annotate it. So, Les Watling, I could swear I heard your voice. And after we do the Metallogorgia, there's a shrimp below it that is the first of that species for the dive. But let's get the Metallogorgia first. Les, you want to tell us about this one if you're on right now?
ROV nav, move initiated.
Thank you, Bridge.
He may not be on.
I guess not.
See him. Okay, fine. Be that way, Les. Okay, so this is in the family Chrysogorgidae, and it's Metallogorgia, perhaps Metallogorgia melanotrichos. We heard a description of this remarkable relationship that the species of coral has with this brittle star from Scott France earlier today. And so we could go ahead and zoom to the shrimp whenever you're ready. We got some really beautiful video of this on a previous one. Now, I think I know what this is, but Mary Wickstein, if you're still watching and near a phone, I wonder if you could tell everybody what we're seeing. And if not, I will.
I'm having a time play here, but that's a front view of the genus Heterocarpus. You've seen them on many of the dives. They're big enough to eat, and they're actually trapped commercially in some parts of the South Pacific. The problem is getting the traps down there and paying to get your boat out to where you get the traps. They're not cheap.
So, Mary, would you even go to Heterocarpus levigatus, or are there enough of the Heterocarpus species out in this area that you'd want us to leave it just as species?
I think you're close. It's tough to tell because the keys start with some fine details that are difficult to see, but if it isn't that, it's something awfully closely related to it.
Yeah, thank you very much, Mary. I appreciate it. It's calling from Texas A&M.
Okay, ready for
I
ride?
think we're good pilot when video's ready. Oh, excuse me, right behind there. Well, we got some umbellula, right?
Yeah.
We did? Okay. Just a snap zoom on it then. That's the C-Pen umbellula. Kind of a productive little stop. Three creatures for the price of one.
Absolutely. We've talked before about how certain animals resemble one another in the same kind of environment. It's always struck me that how umbellula and the stalked crinoids and a lot of other stalked animals basically have the same sort of general shape. You know, there's a stalk, there's a big head-shaped thing with a filter feeding array.
There seems to be a lot of pressure to get off the bottom for a lot of different types of animals.
Yes.
Right, it's coming out.
Okay, we need to see a sponge. I'm getting
on this guy.
I'm getting a sponge itch.
You and your little
Itches.
ones.
Well, there's a star, not a star, black coral.
There's another
thing.
metallogorgia, but yeah, the thing behind it is of interest. And that's Bathypathes, Scott and Tina.
Yeah, it looks like Bathypathes to me, and I'm not sure, but at the base of that metallogorgia, there may have been a sea star, or I just didn't get a good look, and it was in a ophiuroid, but it was at the base of the holdfast.
Let's come out. Let's look at the base.
Oh, this Bathypathes looks like it has two polychaete worms in the center. Those sort of brown things, sorry, you've already moved, I got the 7-second delay. But that's something that Tina's often looking for and is pretty common on the Bathypathes. I'll leave you to the sea star.
Maybe we could just do
Brittle star, I think.
Yeah, brittle star, but there's a sponge over to the left. Yeah, finally. Aha. Okay.
On the base of this metallogorgia, you see there was no tissue. It was black. I'm really sorry. I've got the delay, so I keep describing it after we leave. I'll leave you to the sponge.
Yeah, no, you could disturb me about the sponge because right now I'm blanking on this thing. That is a very twisted Atlanticella. That's my best guess. It doesn't even make any sense. It could be just where it settled or it's something else. But Euplectellida, probably a glass sponge. And Very
Could
unusual.
it be two of
ROV
those that
nav,
we were looking at
bridge,
before?
move
You said
complete.
it was a
Thank you,
lace
Bridge.
sponge with that flowered out at the top.
Well, Roland, that is a superb guess. Because I think you're right.
Maybe three of them here.
Yeah, it's possibly even three of the Atlanticella, really small ones. You have one, you're looking right down the funnel on the left, and then you get a side view of one of these in the middle. I'm not sure what's going on. Maybe it's something else altogether over in the lower right, but I think those two, left and middle, are definitely Atlanticella. That's very, very astute. Thank you very much, Roland,
See,
for
we
that.
pay
Come on
attention.
out.
We listen to you guys and learn along with you.
It isn't just a matter of paying attention, Roland. It's interpretation as well. That was excellent.
Have you at 75 degrees, my camera pilot?
Pushing out.
Co-pilot, the ship finished moving a minute or two ago.
Copy that.
Well, the train is slowly getting steeper. And corals are not getting denser.
The floating jelly. It's inside the lens.
Probably too fast.
Yellow top right.
Yeah. Snap this real quick. Just nothing on it. Alright, coming out.
Dead stock. Pilot, I'm feeling itchy for a rock. There's kind of a nice angular one. Do you think that is too
We
big?
might
I
have to
don't
push
see the
ship
lasers
back
yet.
if you want to rock.
There they are. Would that fit in?
So I'd be at 7.5 right now.
Not?
Well,
Probably another couple meters to go.
Yeah.
not in the greatest position to grab one.
Oh, okay, we can wait.
If you don't mind waiting.
No, not at all. It just
Okay.
caught my eye. That's all.
Alright.
It's one of the few that wasn't completely rounded.
Yeah, roger that.
Yeah, if you're good, pilot, I have
There's
you
a rock
at
attached to
8-0
that
now.
coral at the top of the screen.
The top of the screen. Yeah, maybe we could take a look at this coral, pilot. I think this is different. And this could be one of Sako Matsumoto's beasts.
We did actually see a plexaurid earlier.
Did you? Well,
ROV
I
nav,
missed a lot
bridge,
at lunch,
move
don't I?
complete.
Thank you, Bridge.
Was it this one, like a yellow one
Yeah,
like this? It
think
was.
so.
Yeah,
Oh,
already were.
okay.
Yeah, because Scott went through it in detail.
Oh, okay. Well, then we could pass.
I don't know. Is this a plexaurid though?
Sure
At
looks
least look
like
at
it
the disc.
to me.
Of the Fulfroy.
Yeah, I think this might be the one that we saw earlier, Chris. Chris.
So you're saying it wasn't a particularly startling spot, was it, Chris? Okay.
A startling spot.
Startling. Yeah, I mean, I spotted that coral, but I guess you're thinking.
Oh,
Okay.
sorry. Yeah.
Okay.
Nice rotation.
Alrighty. That's alright.
See, I'm just letting the current do that, Sean. Very little actual thrust.
Oh, yeah. That's nice.
Well, this
Just letting me take
could
it.
be similar to what we collected a few days ago.
Have you
Did
done this
anyone
before?
mention how black this colony
First time.
turned in the ethanol?
Alright, come on out.
That's,
I did not.
yeah.
I did make a note in the event log that
Pilots, I'm playing with
if you notice
tracking a
here,
little bit.
the
It'll be
yellow
coming on and
color
off.
is in the soft
Copy.
part of the tissue of that polyp. It's not in the sclerites, and so my prediction was that if you did sample this and put it in ethanol, it would leach the pigment and turn the ethanol black.
Good point, Scott.
Alright, Chris, if you see a rock up here,
Yeah, you're in a better spot now.
Let's see where are the lasers. How about above the lasers right now, that kind of angular one? Go further up. And a little bit further. That might be a little big, but keep going. Maybe that one above the lasers? Or I don't know how angular it is.
that's a little big.
That's a little big,
I
okay.
mean, there's like
How about this
this
one?
guy right there. It's kind of angular.
This one right here? Okay.
I don't know. I mean, I'm just...
Can we do a zoom on it?
Sure, come on in, Roland.
Yeah, that's kind of a nice angular one.
Alright, come on out. Let's get set up here. So, would we have a rock in port?
Rock in starboard. And it's,
yes, a rock in
so we have to go port. I was told there's no more room at all on starboard.
All the way full.
I can bring that wing in.
Hydraulics up?
Yes, sir.
Alright, bring the... Well, let me see if this is loose first.
Copy.
It is. Oh, that's a nice one. I like that.
Ready
rock.
for wing?
Yep.
John Smith, if you're watching, I hope you like that rock too. I know you responded on the event log a little bit earlier, but
Grab a soft squishy, I can't open the box.
I'm the opposite. Great at the box, can't do anything else.
Do you want me to
I
put
don't know
a
if you
camera
heard
angle up?
that. Yeah. It's actually that C-pen that's doing that. There's a little bit of goo from the C-pen on the claws. Oh, nice move. Nice move. It's all in the wrist.
Yep, that is...
It is.
Absolutely.
that is
There are
absolutely
3
true.
joints in that wrist, and that's most of your maneuverability.
John Smith. Yeah, that's the one, John. That's what we were going
Alright,
for.
You want to tilt up on my camera,
Yes.
and then we'll take a look at this.
Tilting
Alright,
up.
come on in, Roland.
Do you want me to put lasers on it, or do you want...
Yeah, we can put lasers on it. Lasers. Alright, come on in.
That's a nice rock. All right, Nicola, right now, let's up the game here. How much does this weigh? Oh, wait, there's something on the
Sea-Star?
bottom here. That's
Sea
an
anemone,
anemone,
I'm betting.
yeah.
Oh, and a hydroid. Or something off to the
Yeah.
right.
Okay, so we've got a little tubularid hydroid maybe and a sea anemone. You'll have to
It's an
see
enemy.
if it comes up with those.
I would have thought that
I
was
was
a
gonna say,
starfish.
how much does this rock weigh? We have a guess the weight of the
Alright,
rock
come on up.
before we actually weigh it every night in the lab. And right now it is 6 for Matt and 5 for you and I, right?
Yes.
Alright,
Okay.
come full wide.
Well, Chris, are pilots allowed to play that game? Because we can actually kind of feel what it weighs.
I think you could. So you guys got a guess?
No, I turned off force feedback.
Oh, never mind. Josh doesn't like force feed. It's a personal preference thing. But
Okay.
Alright, we can close.
Thank
Port
you very
wing
much,
coming
pilots.
out.
We've got our rocks for the day.
Nice close.
Scott guessed on the first rock collection 15 kilos. And I don't remember whether that was right or not. That was a while back.
I wasn't here for that one, so.
Alright,
Nice.
secure. Alright. We got the waypoint now?
Yep.
Alright.
All right, onwards and upwards. I wonder if we could pass by this bamboo this time and see if we can get a little further upslope. I'm not sure how our progress is. Maybe we can get an update from Nav on how we're
Can you
doing
turn
here today.
hydraulics on for a second?
On? Yeah.
Let's just do a snap here.
Sure, we have moved about 370 meters. We have 350 to go. We need to leave the bottom 2 hours 40 minutes, which requires a speed of 0.07 knots, which we should be able to make.
Yeah, we're good on that ground.
Thank you very much, Nav. I appreciate it. I assume all of you heard that. We're making a decent pace here. We're cruising along, getting our to-dos, close-ups and specimens, and we're in pretty good shape for reaching the top of the cone as well.
Yeah, Nav, we could probably get a move in now, too.
ROV nav, bridge.
Go ahead, Bridge.
I'm getting a bit of a wind shift. I'd like to bring the bow to starboard 5 degrees, please.
That'd be fine.
Coming right.
Yeah, co-pilot Bridge just called down. They're going to adjust their heading to starboard, but we can do that in a couple minutes.
Yeah, Copilot copies.
I wonder what it was that changed from the area we were in to what's happening now. The surface looks similar, but there were just more animals.
I have a hypothesis.
And
It was that thing I was talking about earlier on, that maybe there's a little bit higher current flow around the base of a pinnacle than up slope a little bit, and that's kind of where we were seeing the corals, albeit they weren't in tremendous density, but now they've pretty much petered out. So maybe
so there's no current up here?
or not as much acceleration.
Come on in.
Another bamboo whip. These guys are going to compete with the red crinoids for the most abundant animal, I think, at this site.
Same kind of general morphology, stock.
Right.
Yeah.
But different species, as Scott was quick to note.
Yeah.
And there's another plexaurid up there, another yellow one that we've seen a couple times already.
Just get a snap as I come by.
Yeah.
Co-pilot,
The
does
same one.
010
And
still
anemone,
sound good?
This one does. On
Yeah.
the right-hand
Yeah,
side.
it's all good to me.
As soon as they're happy with the new heading, we'll do that.
Yeah, copy that.
Right.
ROV nav, heading move complete.
Thank you, Bridge. Are you ready for a ship move?
That we are. Go, what'll it be?
That's range 30 meters, bearing 010 degrees, speed 0.2 knots.
What do we got here? Is this just a small bamboo, and there's a round thing on
Range
the rock?
30 meters, bearing
See them,
010,
but
speed 0.2.
Yeah,
Copy.
Yeah.
Coming up.
the coral is a coralliid, I think, too, but maybe a different type.
Come in tight. Alright, let's see the coral. Come on out. Alright, come on in.
No, I think this is a different species of coralliid here. You can notice the very white branches with the pink polyps. And I don't think it's just
ROV nav, move
morphological
initiated.
plasticity or different
Thank you,
colors
Bridge.
of things. I think
Co-pilot,
this
we
is
are
different.
on the
This
move.
may be something
Copy that.
that we're tentatively calling Hemicorallium nearlauense, that has been sampled up in the Northwest Hawaiian Islands. Does have that distinctive white branching, and it's a little bit wormy too. You can tell by the holes in the branches. Those are worm burrows in the branches, and it's common in Hemicorallium. And is that a little ophi down there on the left? And you can see the closed up polyps there too, kind of nice.
It's worth noting, Chris, since you pointed out these sort of wormy tubes, that there are polychaete specialists that live associated with corals, and the one genus that I remember
Say it again,
so
video?
distinctly, because its name is so perfect, is Gorgonia
my fault.
polynoe, which combines both Gorgonian for the coral and polynoe.
Video, we're
for
reading
the other one.
3.08 degrees. down
That's a
here.
nice name. Yeah, thank you, Scott.
Thanks.
we have lasers on?
And actually, Scott, you just
Yes.
reminded me that
Yeah,
we
let's get
did
lasers.
collect a hemicorallium. Well, I actually collected a couple on this cruise, and one of them we did pull off a worm from the specimen up in the lab, when it got up in the lab, and we've got that in ethanol right now.
Let's get the small one in the back too. Just snap in.
Yep, there's a very young one, perhaps of the same thing.
Yeah, it's got a little pearl star on it.
Yeah, it's already got his associate. It looks like a young brittle star too, right?
Yeah, I think so. Well, yeah, I think it is a young Urialet up to. Is there any more zoom?
Any more zoom?
Max zoom.
All right, I can get closer.
Oh, okay.
Coming out.
Really far out. I can't get any light on Sirius.
All right, come on in, roll. Right, go ahead.
Yeah, that's the juvenile of that snake star. That's on the. It's been seen on the other
Any more?
ones.
It's max zoom.
Yeah. Cool. That's, I think, just what I needed. Thank you.
Right, just come on out.
Yeah, probably that's close to your
Nav, what's the?
leash limit right now.
Go ahead, Watchley.
Yeah, hi Dave. We had a question in the event log that I'm just seeing now. So what's our off bottom time today?
15:45 local. Now that's in. It's in 2 hours 30 minutes.
Okay, 15:45. So that's 2 hours. Got it. 2 hours and 35 more. 2 hours and 35 more minutes. That's answering a question in the event log.
Can we get a close look at the skeleton over here? I'm guessing it is a
Come on in.
devoured trident bamboo coral. And I'm saying devoured because it shouldn't have those branches if it wasn't fully grown. Yeah, indeed. So this is an old one that's mostly broken up. No bamboo coral tissue remaining on it, perhaps some hydroid growth on it.
Yeah, that looks like hydroid growth. The fragments that are, or the strands that are on it. Makes me wonder where the sea star is then, or if there's something else.
Well, who knows? You know, this may have been dead for the last 10 years.
That's true.
Maybe the sea star is long gone and moved on to happier hunting grounds. Which is to say, I don't know how long these skeletons can remain in situ after they've died.
Another Metallogorgia.
Something
Is there
white
something, a fish
floating
in the
at the
water
top
column?
of the screen.
Right at the top of the screen, please. What is that? T-Dog-Nath?
Yeah.
Oh, and there's corals on that boulder up ahead. I don't know if that's on our planned route.
You have a little more leash, but
Could be things
not
that we've
quite
already
that much, I
seen,
don't
but
think.
at least they can see some reasonable sized fans up there.
I can't resist pointing out that the consistency of one brittle star with one Metallogorgia continues for each Metallogorgia we see.
ROV nav bridge, move complete.
Thank you, Bridge.
Well, what do we have here? Is it a sponge and a coral, or is it two corals? Aye. Holding breath. Please, if we could look at that sponge. Is it two corals?
Come on in, Roland.
Oh, it's probably a Metallogorgia again. There we are. No, it's different. Chrysogorgia. Okay. Is that the first? Did you see any of these at lunch?
I don't remember, honestly.
Okay.
Yeah, I do not believe we've seen any Chrysogorgia today.
All right, a newbie. That's good.
You guys want to talk about their associates again?
Well, the first thing I'll say is, so this is in the... Sorry, Chris, do you want to take it?
No, that's fine. You probably know more about the squat lobsters anyway.
I was actually going to say something about the coral, meaning that we've been looking at these Metallogorgia. They're in the same family, Chrysogorgiidae, as the one you're looking here. This is the genus Chrysogorgia. And that genus-level naming difference tells you that there's some fundamentally different morphology. And on this particular one, you can see there's branches all the way from the top to the bottom. And this does have at least a pair of associates in there, these squat lobsters, Chirostylidae. The name Chirostylid refers to the very long arms, translates as a spear arm. And we often see these sorts of crab or crab-like animals, squat lobsters, in these Chrysogorgia colonies, and it may be that a specific species of squat lobster likes to live in a specific species of Chrysogorgia. I'm having a close look at this coral, but you can see that some parts of the branch seem even more golden than others, and I'm wondering if there's something else going on there, or if that's just a trick of the light. It might just be a trick of the light.
I don't know, Scott. I was wondering if you were going to mention this. It almost looks like it's saddlebags or something, but I can't really tell. Or maybe that's wrong.
It belongs to a genus called Urotychus that characteristically lives with your Chrysogorgias. However, the species of this group are totally a taxonomic mess. Nobody knows for sure whether this is an already described species or whether it's an undescribed one. All I can tell you is that particular morph has shown up in just about all of the pictures we've seen where there's been a squat lobster associated with the Chrysogorgia, so it seems to be an obligate association. But what the crab is, we're just going to have to stay tuned.
Mary, do we know when we see a pair here, are we assuming that one's a male and one's a female, or are they both females hiding out in there brooding their eggs?
We think they're male and female, but the trouble is that the specimens we have are disassociated, so we don't know if they came from the same coral.
That is a problem.
Mary, you might be happy to know that we've got some other squat lobsters on this cruise, and we may have one of these as well. I just can't remember, but we have definitely collected a number that were associated with some of the corals we collected.
And I'm just itching to see them.
Your laser's back on.
All right. Anything else here, Chris?
I think we're good. Thank you very much. All right, we are still... Oh, yeah, we could take a snap zoom on the sponge. I don't think it's one of the ones that had the critters in it that Mary was interested in, but let's take a look at this. Looks like it's a Poliopogon.
Come on in, Roland.
It's a glass sponge. Yeah, and these this is a different species we haven't seen for a while. We saw it earlier on this dive. It's got the squared off top. Oftentimes you see them scooped out in big kind of bowls on their side shapes. And this one is sort of columnar with a squared top. So this is still in the genus Poliopogon. It's in the family Pherodimatidae. Glass sponge attaches to the substrate with the grappling hook modified spicules called basalia. And these are very cool animals, at least in my opinion.
Can you sort of define for us what a Poliopogon is, layman terms?
Poliopogons, yeah. Our big problem, we have to use these names, and we're not just trying to act or sound, you know, hoity-toity. We don't have a name for the sponge. There is no common name. Just the only common name is glass sponge for dozens and dozens of these things. So we have to use the scientific name.
Okay.
But
when you get
I don't actually know what that particular name, the Latin root, is referring to. Sorry, I can't answer that question any better, Roland.
I'd like to throw out a stump scientist every now and again. Keep you guys growing with your knowledge, too.
You know how old I am, right, Roland?
27 at last, I heard.
These stocked crinoids are just getting bigger as we go uphill.
That's a good sign, and I see a few things upslope. It looks like there's something up there, and it is getting steeper.
Coming up at you, Sean.
Yeah, copy that.
That's something white on the seafloor? That's just a
base, I think.
Base?
Let's snap on it, just make sure.
Yeah, that's what it is.
Or a very well-disguised sea star.
You know, that would be a good camouflage, wouldn't it, Pilot? If you look like a stump.
Yeah, coming up.
You might have a problem shooing away all the hydrozoans and foraminifera and things. Want to settle on you, but...
Snap this guy as we come back.
Chris, I was just
Is that
having
a
a look
big
at my
snail on the
dictionary
bottom
of word roots
below
here,
this?
and I see that pogo in Greek means beard. So I wonder if polyopogon means many beards, referring to the many spicules
That's
that attach
different.
it to the base.
Thank you, Scott. And what do we got here, Chris?
ROV nav.
Looks like,
Go ahead, Bridge.
oh, that's curious. So it
Now
looks like a
the,
snail,
wind is
but it
backing on me
has,
again.
it has,
I'd like to come to port.
oh yeah, these are those zoanthids that
That'd
we
be
saw
fine.
the
Go
other
ahead.
day.
All right.
So zoanthids are cnidarians that look like sea anemones, but these grow on mats, and it looks like it's somehow managed to kind of expand or settle onto the shell of this snail. Bummer for the snail.
Well, is it bad, or is that like protection?
Oh, it could be. Yeah. It depends on what the ultimate objective of the zoanthid is, I suppose. I mean, if it's just being moved around and we're doing sort of a win-win associate thing like with hermit crabs, but...
Excuse me for chuckling, but boy, if I wanted to move around, I wouldn't settle on a snail.
Yeah, well... There's zoanthids on mats. What do you expect?
And also, thanks, Scott. I just stopped to
something pink
thank
off that
you
rock on the
while we
far
saw
right
the
of it.
snail.
It just caught
I
on
think
the
you're
corner of
right
my eye.
about the beard and referring to the attachment of the sponge and the root of the word
The
polyopogon.
top rock,
So thanks
top
very
edge of
much for
screen.
that.
Yeah, no problem. I was just wondering about the zoanthids on that snail there. Maybe the zoanthids are waiting for a hermit crab to come along and take over that shell.
Now you're talking, Scott. Now you're talking.
Yeah, come on in, Roland.
It's like planning for retirement or something. They're long-range planning. So this is an anthomastus or an anthomastus-like animal. Could be pseudanthomastus.
Got a worm crawling on him.
Tina, I don't know if you're awake. They're in Moscow. She's with the Shirshov
Excellent.
Institute.
Oh, it looks like there's a worm on it, too.
Yeah, there's
A
something
little
on it.
polychaete or something. Yeah.
Hey, come on in.
It's max zoom.
Oh, it's Max. Okay.
I suspect that's sufficient.
Just get a steady shot here for a second.
Yep.
All right, when you're good, Roland. Something on this guy.
Ooh, is that
Oh, there's something on
that
it,
hope?
Babu. There's
is
hope.
that hope I see on the
No,
horizon?
negative, Chris, negative. Ooh, look